talk? Is that cool? Fabulous. Excellent. I'm very vaccinated. Hi, I will try to clip 15 minutes from this talk, but it'll still be lots of fun and worth the wait. Welcome. I'm excited to tell you about the work that we've been doing. The highlight will definitely include ammonites and many other things. But before I wanted to go too far, I wanted to thank the people that I work with, and everything that I will be showing today was made possible by the wonderful students, staff, and faculty that we have here at the U. I especially wanted to highlight working in the marine paleoecology lab, Zach Wistor, who defended successfully his dissertation yesterday. Woo, yeah. Undergraduates Jane Baer and Jenny Hamilton and Peter Maxiner, who finished his master's in the days of COVID and now is doing a PhD at USC in Southern California. And the completely separate lab that I tend to run is the Ammonoid Motility Modeling Laboratory, where we have Yoonji Choi is an engineer working with us. Olivia Jenkins is actually a video game designer working with us. Undergraduates Brittany Hoskins and Casey Crawford. And Nick Hebden, who defended his PhD and now is doing a postdoc in California. And Michaela Heber, our newest PhD student in the lab, and our wonderful postdoc, David Peterman. So these are the people responsible for a lot of the big ideas and executions and experiments that I'll be showing you today. The really fun stuff is going to be the robots, which I'll save for the end. These are actually in the Bowen and Bowen pool, where we have, you know, David and I were actually lucky enough a couple weeks ago to go to the Triassic-Jurassic boundary in Nevada that I'll tell you more about, and look at some of these fantastic large ammonite fossils that we are also trying to figure out how these animals lived, and what better way to do that than to actually try to be Bond movie villains and create robot ammonites. But before I get into the specifics of the work that I do with these wonderful people, I wanted to pose the big question: what can we learn from fossil ecology? I'm a paleoecologist, so rather than just, not to discredit, but a lot of work has gone into using fossils, particularly ammonites, for biostratigraphy, for biodiversity studies. And what my research team and I tried to do is put together, how did these animals actually live as part of an Earth system, both in the minutia of could they move and hunt and what was their life like day to day, to the larger, how did this influence giant biogeochemical cycling, so that I can inform other people who study that. Now, if we want to think about this from the sort of, you know, I was thinking about Tyler joining us today and thinking, well, they're applied things and fundamental things, right? You know, we're talking about robotics, but we're also talking about the Earth life systems. And if you want to get a little more cynical, which I usually am, this is kind of the spectrum from the practical things to the kind of woo-woo things. And on the practical side, one of the reasons I love the research I do here at the University of Utah is, I think my undergraduate students can attest, it gives you lots of practical skills: statistics, computing, problem solving, and getting into the hydrostatics and hydrodynamics. We start getting into more of the theoretical, where we start getting into mass extinctions and Earth systems. And I really wanted to highlight here, I put it in cursive, imagination, because I think for paleontologists, sometimes we end up in a dearth of imagination about what past life could be like, and we end up discounting possibilities or failing to ask questions of the Earth life system that we really should be thinking more critically about, but we lack the imagination to do it. So I'll put that on the woo-woo end, but it's still very important. So questions that I will be asking today all center on: how did past ecosystems differ from today? What features are unique to that particular time and place in the past versus what features are enduring fundamental essence of how Earth and life work together? And I decided that to bring us all of the things I'll tell you about today together, I would like to bring you to a place and a time where there was a global mass extinction. And before this global mass extinction, you might have gone snorkeling and thought that the seascape before you was not that different from today. We would have coral reefs, we would have corals and animals on them that were very similar to the things that we snorkel with today, the things we put on pasta today. Clams, snails, you know, shrimp. But some very nasty things happened. We are going to talk about early in the Mesozoic, the end of the Triassic period and the beginning of the Jurassic period. This is an interval when effectively Pangaea, which had been assembling for quite a while now, decided it was done to anthropomorphize. And Pangaea started ripping apart down what would one day allow the Atlantic Ocean to form. But these giant sweeping influxes of flood basalts as dikes and sills and flood provinces that now are found as rocks in South America, in Africa, in Eastern North America, excuse me, are all from this interval of massive recurrent volcanism that is credited with changing Earth's climate, atmosphere, ocean chemistry very geologically rapidly. Observations from plant anatomy and from the chemistry of ancient soils help geochemists interpret that the carbon dioxide in the Earth's atmosphere was much more than today. In the end Triassic, it would have been in the hundreds of parts per million, and in the early Jurassic, perhaps the thousands. So either way you slice it, we're talking about a warmer, wetter climate going into the Jurassic, and we're talking about a global mass extinction that impacted life all over the planet. And I will be focusing on sea life today because that's my jam. Now, I'm highlighting here with these red lines the sort of 5 big mass extinctions that paleontologists like to pay attention to. And in the background, I have a diversity curve of marine life through the last 500 million years. If we divide this marine life into categories, and we tend to say, I'll try to use my mouse for those of you playing along on Zoom. I can't see my mouse, so I won't. If we divide this into categories, I'm highlighting with the black line things credited as the Paleozoic fauna, things we do not eat on pasta, brachiopods, crinoids. And then the modern fauna in blue here, things we do eat on pasta, clams, snails. And if you watch this blue line, our interval of key interest today, the Triassic-Jurassic boundary, among the reasons this mass extinction is interesting is it absolutely destroys the biodiversity of animals with physiology and ecological system behaviors that are a lot like what we have on Earth today and that we value today and that we eat today. The clams and the snails bite it. And diversity in the marine realm of marine animals stays very low for a while. And so our task today will be to look from this Triassic world into the Jurassic world, and looking at this Jurassic world and trying to look at the rocks and fossils and say, what can we see about how this system was different than it is today? And what of those differences are because of fundamental underlying features of Earth life systems, and what are kind of one-off fluke, wacky early Jurassic stuff that's, you know, fun, but nothing to write home about. I'll still write home about it. I love the Jurassic. So thinking about this transition, broadly, I will tell you, we are going from a world with many coral reefs to many probably very stinky sponge expanses, and from a world with many small ammonoids of different shapes to a whole lot of really boring ones. Now, a funny thing about working on the Triassic-Jurassic transition is that fossiliferous rocks from shallow marine settings, very hard to find. People have been working on it for a while, so Europe's pretty well covered, but around the rest of the planet, it's still pretty difficult to find a cohesive marine section that can tell you the story of this global change. And one of the places that I have loved to study this is in modern day Nevada, which was on the coast of Pangaea at the time, and in modern day Peru, which was on the coast of Pangaea at the time. So we'll visit both of those places briefly to think about what the ecosystem was like. This is a hillside out in Nevada where I did a lot of dissertation work, and David and I were there 2 weeks ago with Randy Urmis and the crew from Reno. It was very exciting to kind of visit back home. But among the things that I bumbled into when I was trying to find clams and snails and failing in graduate school, in these earliest Jurassic rocks, what I was finding instead were sea sponges. Sea sponges should not fossilize. There's a picture that I took on Brenda's camera when we were in the Bahamas right before COVID. And little sponges should dissolve. They don't even have tissues. Their cells should die, they should dissolve, and their little spicules should go scattering into the sediments, end of story, goodbye. But finding body fossils of sponges and then finding enormous bedding planes, this is a dip slope. So it's bedding plane after bedding plane after bedding plane of body fossil sponges. That's not supposed to happen. You're not supposed to see that. And they're in situ, just like they got Pompeyed when they died. So measuring many strat columns through this interval and trying to look at, okay, not much to see here in the form of shelly fauna. If you wait until the rocks that represent a couple of million years after the mass extinction, then you get corals and snails and clams. And they are evident in the thin sections, and we can put together this picture of this system having sea sponges right below fair weather wave base, dominating seafloor habitats locally. And this is important for two reasons. One, sponges aren't usually shallow in today's world. Today, if you have a whole lot of sponges, it's very cold and very deep. But these sponges were right up to fair weather wave base. And our interpretation of this is that they ended up getting sort of finally dethroned and deposed by the onslaught of a carbonate factory that generated a new shoal that was too unstable for the sponges to live on. Well, this is well and good as an interpretation that, oh, in the early Jurassic, we had some sponges, you are welcome. But this is so far a local story, which is why it's nice to go to the other side of Pangea's coast to the south by going to Peru, where the Pucara group is spread across the nation, is very important for gold, silver, and polymetallic ore mining, which means that people have studied the absolute detail out of these rocks so that I can waltz up and say, hello, Silvia Rosas, who at the time was a chair of geological engineering and mining engineering at the Catholic University in Lima, and say, hello, may I look at those thin sections from your dissertation? Will you take me in the field and show me where you got these three that have sponges in them? I see there are a lot of cherts in these rocks. I see these rocks represent the Triassic-Jurassic boundary. She did her dissertation on these rocks a generation before me, allowing me to just kind of waltz up and get the cream of the crop of the Andean Triassic-Jurassic boundary sections, which was very spectacular as a finishing grad student to have these opportunities. And then together with the undergraduates who've worked in my lab on the microscopy and Peter Maxiner, who finished a master's with us, we've been able to put together these kind of broad strokes. So let's see, the Triassic-Jurassic boundary is in debate, but it's somewhere around here. And then these great big orange stripes are rocks that are replete with fossil sponges. We can clamber around on these rocks and look at them, and they look kind of horrifying. If you saw Zach's talk yesterday, that was sort of a theme of his talk, was how miserable these rocks are to work on. But when you put them under the microscope, they're really quite remarkable. In the rocks are completely composed of the little microscopic bits of dead sponges. And if we put together, these sponges are everywhere. They're in the fine sediment, they're in the medium sediment, they're in the coarse sediment. These sponges are making different kinds of configurations, and this is some of Peter's work that he's about to publish, putting together how what he can see in the microscope contributes to what he can model about the ecosystems and the animals that were living together on the seafloor when there were clams and snails, and when the sediment was being flushed with detrital sponge bits. So this allows us to kind of put together this picture of a coastal setting where sponges are not only living in certain pockets, just the debris, the garbage, the schmutz, the sediment everywhere is filled with sponge matter. And this, on one hand, sort of substantiates this idea that in the early Jurassic, these sea sponges were doing really well. And it's very different than the underlying Triassic rocks. And again, all of this work has been done with me since I moved here. A lot of the microscopy has been done by Jane and Jenny, who have, and you know, this was Trogador is a microscope I bought with startup. So that has been a boon to many people in the department. I want to take an aside now. I'm focusing most of this talk on the Triassic-Jurassic interval. But because of our sense of imagination, because of this woo-woo direction of our questions, because of this idea of, wait, sponges shouldn't even be found as body fossils typically, then you're telling me you have these large expanses of body fossils. Is that just because there was a global mass extinction, or is that some fundamental thing that sponges typically do in the fossil record? And if you just try to look for it, you'll find it everywhere. So I put together a review for Paleo 3 a few years ago, looking at known sponge rocks of the world, because it does happen sometimes. And a fun phrase for these is glass ramps. A carbonate ramp makes carbonate. A glass ramp makes biosilica. It's very different from a siliciclastic environment, which would be quartz and feldspar, detrital, sands from, say, mountain ranges and rivers and beaches. This is completely different. This is biosilica. And our contribution to this, these have been documented in the literature from the Mississippian, from the Permian, I'm adding the Jurassic, and also from the Eocene. Now, conveniently, the Permian rocks are right next door. If you drive out to the West Desert, just northwest of the Great Salt Lake, there are these beautiful, we living here in Utah, we're on the transition from the Rocky Mountain to the Basin and Range. And these small, scrappy mountains that you can drive out to on your way to Nevada, as you pass Wendover and there's small mountain ranges, you could scamper to the top of one of those mountain ranges in a day, not a big deal. And this one, here's a suburban for scale. And our former longtime master student, Shauna Hood, took this picture. This is suburban for scale. I like to call this, say, Helms Deep. This cliff here is part of a formation of late Permian rock that is about 1,000 meters thick of almost pure chert. That's weird. And what it makes in some cases are these kind of giant steps. I dragged Peterman out there to his confusion in October. There was not an ammonite in sight. Okay, I'm not over there. I don't exist if I go over there. I'm over here. These very large steps, even closer, these very large steps are each made out of actual vase-shaped or like stacked pint glass body fossils of sponges, which are completely bizarre. And Zach gave his dissertation defense yesterday all about how these facies fit into the broader regional context, how they interfaced with other animals, and put together his model from microscopic hand sample and outcrop views. Within these sponge steps, we find here and there outcrops of what I would just call brachiopod banks, right? It's the Paleozoic, most of the shelly things you would not eat on pasta, right? And these brachiopod fossils that I'm showing, these sort of big orbs here, these are brachiopods that are so tightly stacked and so closely in situ. You're getting a snapshot of them living together, which is like paleoecologist's dream. They're not all scattered and jumbled together, because when you look at them closely, here is a bryozoan colony still attached to the shell of a brachiopod. So this is some in situ built up rock sized, sort of refrigerator or deck edition sized rock bodies made up of brachiopods. So what I would like to present as the sort of overarching, what's going on here with sponges? What of this is enduring versus some fluke from the early Jurassic that I'm interested in, is the idea of state switching. Those of us who grew up on the California coast or researched out there are familiar with the woe is me about the lack of sea otters, meaning that sea urchins spread out and decapitate all the kelp forests. It turns out that for 1,000 or 2 years at a time, you'll have mostly kelp forests. 1,000 or 2 years at a time, you'll have mostly urchin barrens where there aren't so many kelp forests. Each of those states is a stable ecosystem. And you don't need a giant excuse to switch. Each of them is self-sustaining and can stay in place. And an interpretation I have from all the sponge work we've been doing is that the carbonate setting and the sponge setting are each alternate stable states. Either one of them could exist in this climate, in this setting, on this coastline with these nutrients. And once one is established, the other can be a bit role. They can get, best supporting actor, but they are not necessarily doing the making of the sediment. And so this idea that I've taken from modern ecology and tried to apply to reason with, well, what about larval recruitment? Brachiopods they look like clams, but they taste terrible. They prefer to recruit from larva onto an adult. That's a nice place to settle. That's a nice shell to settle on, or a dead shell. It's a nice place to settle. If you try to settle on a sponge, they get trapped and you'll slowly dissolve and it'll eat your organic matter. It's not going to work. So the idea is that you could have a setting where there are brachiopods and sponges come and go, sponges come and go. And as long as the live sponge cover is fairly limited, you will be continuing to maintain a brachiopod bank. Once the sponge cover of live material is so expansive that your recruitment plummets, you can switch states into a sponge state, and then your brachiopods are really out of luck. And it's very difficult to establish that kind of gigantic coastal system that they once enjoyed. There's still pockets of brachiopods. And Zach's dissertation work that he showed yesterday showed when you find the pockets of brachiopods and bryozoans and crinoids and what have you, it's the same animals. They haven't fundamentally changed. It hasn't altered the course of evolutionary history. It's just different players taking their turn on the stage. So this sort of state switching interpretation gives us a way of thinking about these sponge rocks of the world that they aren't exceedingly common, but when they happen, this might be a place where there's a role for animals and ecosystems and interactions to really be shaping the rock record. And yet, for the earliest Jurassic, I'm still going to blame that on the mass extinction, because I think that wiping the slate clean of many of your biocalcifiers, which particularly aid it in that giant climate change, can actually be triggering that more short-lived event. So it's one thing to study mass extinctions and say, oh, I found something. It must be because of the mass extinction. But part of what I've tried to do with looking at how animals make rock is to step back and say, all right, well, sure, lots of sponges. But is that a thing that happens all the time? Well, it happens sometimes. And in this instance of the early Jurassic, I do think that the context of the mass extinction triggered and initiated and allowed those sponges to settle. And the final 2 million years later, resurgence of carbonate production removed the sponges from those coastal habitats. Now, the thing I want to turn to next is thinking about what's in the water. Let's focus on the same time interval, the end of the Triassic, the beginning of the Jurassic, the aftermath of the mass extinction. But let's focus on these animals that are swimming or drifting or creeping or lurking, right? We've established we have some sponges in the early Jurassic. We've got this whole landscape of sponges. And then we find fossils of ammonites, these squid-like animals that had a seashell. And we don't really know what they were doing. Were they interacting constantly with the benthos? Were they eating maybe snails, something that cohabitated with the sponges? Or were they minding their own business? And for a couple of million years, really presenting a distinct system between the pelagic open water setting and the benthic setting. How tightly linked were those things? Well, if we want to know the ecology of these animals, we can use clues from their living relatives. And all of the work that I'll be showing now is part of what has become known affectionately as the AmLab, the Ammonite Motility Modeling Laboratory. And we have engineers, we have 3D modelers, we have undergrads, we have Nick, we have Dave, we have Michaela. And David and I were actually at New York Canyon in Nevada, looking at the Triassic-Jurassic system just a couple weeks ago. And we were doing the time-honored work of sitting on this exact hillside saying, I found a Triassic one, I found a Jurassic one, I found a Triassic one, I found a Jurassic one. We sure know we're at the Triassic-Jurassic transition now because we found these biostratigraphically valuable ammonites, all right? Ammonites are squid-like animals, they're extinct. Their seashells are found all over the world, and they're really good for telling time and space. And the early Jurassic rocks are filled with huge, boring ammonites. The average ammonite for the last 500 million years when they were around was the size of a golf ball to tennis ball sized. But these things are dinner plate to bicycle tire sized. These things are huge. And when I say boring, I mean their shape is almost always like that of a coiled snake. And in the rock record from all over the world, from everybody who names and categorizes these things, they are this coiled snake shape all around the world for a couple million years. Many different shapes were present before the extinction. Ammonites are actually kind of rare going up until the extinction. And while a couple of lineages did present these rather streamlined, who has my other toy? These more streamlined shapes were present, but this boring coiled snake shape became abundant, diverse, and all these different lineages, everybody's doing it, all these different lineages continued to produce this coiled snake shape. And then some of these things got really big. So an obvious question, ever since I was a youngster out there trying to hit rocks with a hammer and see why no clams and snails were coming out, because I didn't know about the sponges, was what on earth are these ammonites doing? And now, a decade later, we have the tools to ask this question. Now, one of the ways we can think about this by comparing them to modern animals is to think about jet propulsion. The chambered nautilus, which is distantly related, is still alive. It jet propels. Squid, which are more closely related, jet propel. And the animal also has, David made these 3D prints, which are amazing. The animal has a buoyancy apparatus of air pockets, and then the body is stored in the lower chamber. So this allows it to have some kind of neutral buoyancy, some kind of stability or orientation in the water. And when they jet the water out, they move shell first. Well, even more emphasis now on these screwy snake-like shapes, because if they are going to move shells first through the water, the shell is in the way and it becomes the vessel through which they have to navigate the water. Here's Lindsay, who's at the LA Museum, and Nick and I well, I'm sitting. That's what you do when you're the advisor, trying to use actual fossils to make 3D models. And then we have these 3D models, and also we can make idealized, instead of replicas, we can make these sort of synthetic idealized 3D models, which David has methods for, and also Olivia Jenkins creates methods for us. And then we can put them in actual water flow chambers, and we can put them in simulations. Several years ago, we worked with the water flow chambers in the engineering department and realized that they just did not have the equipment to measure the sizes of effects we wanted to see. So Nick switched to simulations, and I said, sure, Tiger, go get them, because I had no idea how to make this happen. And through a lot of blood, sweat, and tears over the next 5 years, Nick figured out how to use ANSYS Fluent engineering software to model how water would flow around these seashells. Here are some of his benchmarks. So these are data that were published for sphere. Oh no, these are his synthetic simulated data for a sphere in purple and for different shapes of ammonite. This is what Nick generated. This is what he generated for a nautilus that is either large or small. What we're thinking about here is how drag increases as the velocity of water increases. So an animal trying to squirt through the water, as the water is going faster and faster and faster, it's making more and more drag. And this is that compared to published literature. So the benchmarks are working pretty well. The kinds of drag that Nick could simulate are very much on par with the sorts of things that people had measured from physical experiments. So for the next several years, we moved on to simulations. And thinking about these different shapes, sort of shape of a basketball, shape of a frisbee, and shape of a snake, ammonites that are simply coiled, not like these weirdos that must be terrible everything. These simply coiled ammonites, well, they come in every variety between these end members. And we might think, okay, well, if you are more stout, it would be generating more drag as you push through the water. It's harder to push through the water, right? And what we're talking about here for this is I'm talking about velocities with the animal about the size of a golf ball, talking about velocities of 5 centimeters per second. That is about, everybody be a sloth, do it. That's about 5 centimeters per second. Whereas a ½ meter per second is more like a busy street where people are bustling along. That's how fast we're talking about. And now when we're talking about drag force, we're talking about feel in your hand, imaginary, a paperclip, an apple, a bottle of water. Those are the forces. We're talking about that animal facing as it tries to move, either just, sloth pace or people in Manhattan pace. And each of these different shapes generates a different amount of drag, and it depends on what shape you are. Now, we can think about this in the concept of velocity and drag, but we can also use the magic of physics to think about these more fundamental aspects of shape. If the shape is what's making you slow, but the faster you go or the bigger you are, you get a discount on the coefficient of drag. How much your shape slows you down is reduced if you're bigger or faster. So all of our shells, if we put them into this concept of Reynolds number, which is assessing how fast and how big, it is our regime of water flow. And then what your coefficient of drag, that's how much is your shape a pain in the booty. These different shell shapes have a whole spread. The discount is different depending on the shape of shell. So these animals would have faced really different consequences for speeding up, for acceleration, for growth, for reaching a large size. And what's more is that if we look at very similarly shaped animals, the compression overall of the shell, just presenting a kind of narrow margin to the water, really helps you reduce that drag and helps reduce the coefficient of drag, right? But this hubcap that's exposed in the snake-shaped ones, it doesn't really start to cost until you go into these higher Reynolds numbers. And then that hubcap starts to show up as a limit on how much of a discount you can get on your coefficient of drag. If we think about it this way, if you are going 5 centimeters per second, sloth speed, then the absolute value in drag difference that it makes is not big, right? But if we think about these forces as the difference between 1 apple of force and 2 apples of force, if you're trying to go to 15 centimeters per second, all of a sudden that shape and that hubcap that you have showing and the drag that it costs is going to make a really big difference. And this is interesting because when we return to our early Jurassic world filled with snake coil bicycle tire sized ammonites, the question is, what the hell? They're bad at everything. Why? They wouldn't be terrible in low Reynolds number, so fluid flow regimes or small sizes, but they reach large sizes. So they would have to be in large Reynolds numbers. They would have to be in turbulent flow regimes. And it seems like that shape is bad. So the idea that, oh, well, after mass extinction, competition should just be raging on this open field of evolutionary technology. It's not really holding up that speed is the thing these animals are built for. Maybe they're just drifting. Maybe they're just lazy. Maybe they're just bad at everything. So taking this a step further, one of the things that we have looked at that I am now thinking is the most important is that that coefficient of drag, the reason it reduces as you go faster or get bigger is because viscous flow, which is from laminar forces. When the water is, think about trying to sink your feet into mud or molasses or syrup, right? That produces a lot of friction drag on your shape. But as soon as you transition out of laminar flow and into turbulent flow, you can leave that viscous friction drag behind, and then you're just facing pressure drag, which is just from your edge on shape. So we can think about this transition as being one of the most important things for these animals, going from being hampered by viscous drag to shedding viscous drag and only have to deal with the pressure drag. This is a fundamental challenge for these animals. Now, what I was just showing you earlier, I will show now again with this idea that turbulent flow, if you turn up the velocity or if you turn up the diameter, each of those will turn on turbulent flow. The Z that I'm demonstrating here with those values, that's the Reynolds number. That is that, are you big? Are you fast? Are you both? If you go fast or if you get big, you are turning on turbulent flow. And this landscape allows us to then think about animals that are going faster and faster or getting bigger and bigger. And this is really important with these animals. More than clams, more than snails, more than other animals, these animals grow from very tiny sizes to very large sizes. And if they are just sort of hovering in the water and then they want to get anywhere at all, they are then going from 0 to some speed. Again, very different from clams and snails, which snails are not getting very fast. So as an individual animal is doing locomotion or is doing ontogeny and growing, it is going to be experiencing not just, turbulent flow. It's going to be experiencing the transition into turbulent flow. A lot of this work 5 years ago when I was doing it, engineers would tell me, oh, well, the things you're asking don't matter because you're in turbulent flow. You're in high enough Reynolds numbers and none of that stuff matters. You're not in laminar flow. Yeah, but we tend to be in the transition all the darn time with our experiments, with our simulations. So thinking about this and breaking down what I was showing you earlier, okay, so Reynolds number expanse and that discount that you get in coefficient of drag. When these things have, these shapes that basically have indistinguishable consequences when you are in laminar flow, doesn't really matter. But once you get into turbulent flow, that's when these shapes have very different consequences for the animal. And we can visualize these things, the same simulations in the software that are giving us the measurements that we are using for these plots can also give us simulations of how the water is getting entrained along that hubcap or in the area behind the shell. And we can think about that drop off in viscous drag. Now, the question becomes, and this is where we'll finally get to robots, the question becomes, what are the consequences for an actual living animal? I can tell you all I want about these forms that we design for fun, and then we say blah, blah, blah physics, but what are the practical consequences for the animal? And this is one of the last things that Nick was able to do with his dissertation before he had to take off for his postdoc. One of the ways we asked this question was, okay, let's think about animals getting a single jet and then how far they could travel, or 3 jets and then how far they could travel, this sort of thing, right? And so these plots that I will show you are just letting an animal have, and this is all based on simulation data, and Yoonji helped us create a one-dimensional trajectory analysis based on the simulation data and those transitions of coefficient of drag with turbulent flow, because the animal is going to start not in turbulent flow, everything's laminar, and then after a jet it's going to get into turbulent flow, and then it's going to cruise, question mark, something, I don't know. So in this plot, the x-axis is, I'll wait, 60 seconds, how far did you get? After a jet, how far did you get in 60 seconds? And then the y-axis is just thrust spent. All the animals I'll be showing at a certain size, they're spending the same thrust because we normalized them. So here I'm showing the bigger symbols for the bigger animals. If the animals are small, we let them have a jet and see how far they get. They all get about the same distance. And all these little symbols are plotting right on top of each other. And then if we say, okay, well, let's give them a minute. Now they're larger. Oh, they all got farther. Let's make them even larger. I think this is 5, 10, and 20 centimeters, the sizes that I'm showing you. Notice that with these larger animals, that spread between them is getting more noticeable in absolute time. Like the distance, some of the guy at the end there, got, and this is on it should be on a log scale. Yes, this is on a log scale. So this guy got well over 2 meters. This guy got not even 2 meters. If you're trying to escape a predator or move efficiently to the other side of the island because you heard there's a good shrimp buffet over there, this stuff is going to matter. Thinking about 3 jets now, this little guy that took 1 jet earlier, if he gets to do 3 jets, he gets a lot farther for not that much more energy. These medium-sized guys, they get to do 3 jets, they get a lot farther for not that much more energy. Same with these other guys. But this actually tells you, your 10 centimeter ammonite can get as far as the 15 centimeter one. He does 1 jet, he does 3, and they're both getting as far. But this also is showing you that these animals are going to eventually kind of hit a wall where the larger body size isn't necessarily behooving them, or larger body size may allow them to be lazier and just expend 1 jet to get the same amount of distance. The reason I bring this up is sort of a fundamental thing, getting back to our kind of imagination of why would this matter for the animal. Anybody who's worked with marine biology, plankton, stuff like that, ocean animals, when they are babies, they look nothing like their adult form. They have ridiculous metamorphosis that they go through. Think about a caterpillar becoming a butterfly. Almost everything that lives in the sea that's an invertebrate, whether you eat it or not, it has that caterpillar butterfly situation. When they are tiny babies, they're drifting in the plankton so they can get to the other continent or across a sea or across a bay or across a coastline. And these tiny microscopic animals look nothing like the adult animal, except cephalopods. Modern day squid, modern day octopus, they are cute as a button and they are ready to murder. As soon as they hatch out of the egg, when they are done with their yolk, they are murdering other plankton. All cephalopods eat other animals. None of them are vegetarians. They have never figured that out in the last 500 million years. And they hatch ready to murder. These other things hatch not even fully formed, not metamorphosed, not with the yolk, and they are doing something else if they are entraining small cells in the plankton. But the octopus, when it hatches out of the egg, it's a tiny, monstrous little octopus. The squid, when it hatches out of the egg, it is a tiny, fast, murdering little squid. And this then asks, well, wait a minute, what are the Reynolds numbers here? Just trying to grow up. Think about if you go to the aquarium in Draper, you can see a giant Pacific octopus. Body's about the size of a basketball. Arms are about the size of a Peterman. Peterman, stand up and flail. Tyler, stand up and flail. Shanna. Yeah, about like that. About like that. That's about a giant Pacific octopus. There's a whole lot of giant Pacific octopus, right? And it's born, boop, boop, tiny, still murdering, but very tiny. And so thinking about the flow regime that that animal has to experience, it's going from this tiny thing to this huge thing, let alone trying to swim, right? Something like an abalone, they actually have this swimming activity, tiny body, and then they settle down and become luxuriously boring, tasty snails. Whereas the octopus, the second they're out of that shell, they are swimming and murdering. So Casey actually, he's not even a student here at Utah. He just bumbled into the lab and we took him on over the summer. He's a student at Indiana University. And he did this study where Peterman made him the shells, thinking about, okay, well, what if this ammonite was sort of detangling this? What was its shape when it was little? What was its shape when it was medium? What was its shape when it was a teenager? And what was its shape as an adult? And what are the physics there if we run those in our simulations? And I'm showing you baby, toddler, teenager, adult in these colors and symbols. But now let me highlight for you the discount in coefficient of drag is relevant for the baby. As it would go faster or be growing larger, it would have, and what I'm highlighting here is the Reynolds number is relevant to that baby going 1 body length to 2 body lengths. I think that's what I did. So if it is just trying to get out of the way of something else, not going very far, but just trying to get out of the way, we notice that that coefficient of drag discount is applicable for that shape. But if you maintained that shape into adulthood, you wouldn't be doing as well as you could if you switched up and traded up and got a better shape. It's like getting a faster scooter when you have better balance, right? My kid keeps getting faster scooters. She's almost 5. She's ready for rollerblades now. So if we then think of each of these additional forms and sizes that the ammonite presents, just through its ontogeny, it's not doing anything, it's just growing up. But as it's growing up, it is presenting a wholly different physics situation. And this can allow us to think about, wait a minute, how fast might this animal have been able to travel at this size? And could we argue that some of these animals, through their ontogenetic change in shape, are expressing shapes that are getting better and better returns on the efficiency of locomotion? Or are some of them absolutely not getting better returns on the efficiency of locomotion, and they are emphasizing something completely different, right? So this brings us to robots because we want to figure out how we can make experiments that ground truth, these actual trajectory models, in real time. So what David has been doing, we put GoPros underwater. We have 2 different GoPros. So the videos you're watching here is these GIFs, is the view from 2 different GoPros. And then he trains some machine learning to track the points on this model ammonite, which is actually squirting water out. It's not a propeller, it's an impeller. So it's actually squirting water out of its face to scoot along. And this is an image of it giving, we hold it in the little tongs, which also have a light sensing trigger attached to a television remote. So these things are actually remote controlled, and we tell them, okay, 1 jet for 1 second, go. And we let them go, and then they give 1 jet and off they go. And then David is using the software to motion track so that he can figure out where they are in 3D space and how fast they went. We've been using many different swimming pools for our experiments, but the Bowens kindly lent us their pool in the fall. It was very, very cold. You can see I'm all wetsuited up. But this is where it's finally amazing to take these animals that we can see as fossils and then actually be swimming with them in the swimming pool. Here's a schematic David made. These things are amazing. They're 3D printed out of plastic. They have a mobile counterweight inside so he can get the balance just right. They have an impeller to squirt water here, and then the battery and a pack of controls that we situate inside the 3D print. That's me being very happy when he finally gave me one that was working. And so these different shapes, you'll notice they're slightly different sizes, but David has made them all the same volume, so that their ability to push with their soft body should all be the same. And then we can look at how each of those created a different trajectory in 3-dimensional space, and put together from that okay, well, as time was going, how far did you get? These are our very first robots. We are so excited to do more with them. David has about 18 ideas per minute, and so we have to figure out how to actually build the robots in the middle of a gigantic microchip crisis around the world. But building the robots, iterating the robots, putting them in the water has been a lot of fun. The other thing that's neat is I have, I'm not going to say I've been afraid. I'll just say I've been conservative to only be working with the animals that have a nice coiled shell that I can understand that's nice in one plane. But David is a specialist in what we call heteromorphs. This Didymosauros 3D model that he 3D printed, he made from specimens that he found in Montana and put together how they would look. And this is life size. We're guessing about the soft body. We don't get fossils of that. But this thing is ridiculous. And here is a 3D model, sort of like X-ray, showing, okay, well, let's consider this. These round ones that have the shell in a single plane, they would have their buoyancy apparatus and their soft body. And the distance between their center of mass and their center of buoyancy, which is how much water are you displacing, and then where's your mass settled within that space. These guys would be pretty low stability. Think of an astronaut in outer space. They could probably hover until they're ready to jet somewhere. Whereas these things have tremendously more stability. What David has been showing with his models that he's making, so you can actually figure out, boop, 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 what the restoring moment on these things is. And these guys would be like sea Roombas. They'd be so stable with their mass down here and the buoyancy up here, and they'd be so good at turning around in a circle or going in a straight line because they oriented their little spitter right in the middle of their center of buoyancy and mass. So it'd basically be like sea Roombas. These animals, everybody said we're stupid. They said, oh, they must be pre-programmed to go extinct because that's how evolution works, right? Because we didn't know what they were good for. They look terrible at swimming. Well, sure. But if you want to be a sea Roomba, they're perfect. So here's a plot, and we've been able to do these experiments and simulations. And here's a plot that Nick put together that he just showed at GSA. This is some stuff that David and Nick and I are working on to conceptualize, okay, maneuverability or stability, which one do you want? Swimming power or swimming efficiency, which do you want? So this old view of, oh, well, there's just good ones and bad ones and fast ones and slow ones. At the end of the day, for this mass extinction, in the early Jurassic, I think you just needed to be good enough for government work. What are you gonna do? It's a sponge apocalypse, right? So these simple, boring, giant ammonites were good enough. And with that, I would like to thank the Marine Paleoecology Lab and the Am Lab, and I am done. Thank you.